goodness, it is Friday. It is Friday, 4:20 p.m. You know what time it is. It's time to get a little bit smarter. That's what we're going to do today on Because Cannabis, presented by Wayward Media. Check us out, wayward.media. Subscribe to the show if you are watching on YouTube, which we know you are every Friday at 4:20. Dustin Kava with me as always. How you doing, Dustin?
Friday, cold but amazing, but I'm perfectly suited for cold.
Cold but amazing. Our guests don't get cold as much as we do, at least not right now, being from California. We are here in Northeast Ohio. It is a little chilly, and when it gets a little chilly here in Ohio, Dustin, the party, as it were, the after events where people go to mingle and hang, it goes indoors. And I don't know if it's not in my genetics or maybe not in my DNA, but I am not a karaoke guy. Are you a karaoke person, Dustin? Do you go to a random bar and choose your favorite tune or song and sit there in front of complete strangers and hold the microphone and close your eyes? Do you sing karaoke to a song so well that you don't even need the lyrics on screen? Or are you someone who sits back and looks at those people singing and just cringes with
I've never sung karaoke in my life. And I genuinely...
Hold, you've never grabbed... Whoa! Never?
I even had the store sandwiched in between two bars, and all you could hear was karaoke. And still, it's not in my DNA to even. I would never even pick a song. Just even thinking about what my song would be is almost it's making me itch.
I just can't. I gotta be honest with you, Dustin. We learn new things about each other all the time. We've known each other over a decade, but yet doing this show every single week for 60 minutes, talking with each other, having our guests on who are probably sitting in the lobby wondering what in the world they've gotten themselves into. I promise we'll get there. You gotta warm them up a little bit, as it were. You know what? Let's just stop this right now. Let's bring them both in. Angel, Oscar, we're going to talk science. We're going to talk genetics. Doesn't matter. Right now, the most important question on the table for each of you, starting with Angel Fernandez from MyFloraDNA. Are you a karaoke person? And if so, Angel, what's your go-to?
Not at all. I'm like Dustin. Not at all.
Not at all. Oscar, help me out. Are you someone who grabs the mic and just puts some lyrical lemonade out in the world?
Not me. I'm so sorry about that.
Oh, my gosh.
Okay.
I'm trying to change that in my DNA by working here in the, we're working every day in the lab to see if I can change my DNA as well as the plants
to make you a better singer.
To make the music thing into my head. It's not working so far. So
I will say I am not afraid of coming out of my cannabis closet, but my karaoke closet is a whole nother bag of tricks there. BC, you're the only one who has a song. I have to know what is it? What is it?
All right. Well, I'm not gonna blow your mind exactly. I'm not much of a singer, right? I don't have a. I wish I talk about this often. I had some deep, melodic, like golden baritone voice on the microphone. It's very nasally. You gotta just roll with it. Also, can't really sing, can't carry a tune. So I go to the rap. So I'm just gonna put it out there. I have many, many times, if you stroll throughout Northeast Ohio here on a cold Thursday night somewhere, karaoke, you will see your boy, B.C. Wehman, with the microphone, definitely eyes closed, definitely not needing the lyrics on the screen, waxing poetically to Vanilla Ice's Ice Ice Baby. I tear that sucker up. I really do. All right, stop, collaborate, and listen. Yeah, exactly. Brand new invention. Maybe that was the emphasis for you guys. Brand new inventions. MyFloraDNA. We have Angel and we have Oscar. Welcome to the show, gentlemen. Welcome to Because Cannabis. How are you guys doing today?
We are doing well. It's raining here after a long season without rain in California. Finally, we are getting water, and it's amazing. It's amazing.
That's fantastic. Do you guys, I know both of you were originally from Spain, is that correct?
Yes.
And now you're both in California. How is just in general, like, what is that transition like from Spain to California weather-wise? I sound so geographically dumb. I'm so sorry. I don't really know what the climate in Spain is. I feel like I should be better prepared, or I should at least pass like 6th grade geography class to know that. But it's been a long time, sir. I don't remember that. That's terrible.
Yeah. Well, actually, there's not much difference. I mean, if you look at the latitude, we're about the same, the same level. So, well, Angel has been here longer than I have been here for about a year, and I haven't noticed much difference, to be honest. It's
you guys are in Northern California, correct, Oscar?
Yes.
We are located in Sacramento. Sacramento, Davis. Yeah.
So that definitely makes a lot of sense. That it's, you know, same geographical weather, same, you know, yeah.
Correct.
Food-wise, Spain, California, very similar. Demography also very similar. Personalities of people, mix of different cultures. So it's a very, very different, similar country as California.
That is interesting. You know, you had spent some time, Angel Fernandez, with MyFloraDNA, the CEO of the company. We'll talk about what MyFloraDNA is doing with testing and such, but you are someone, you know, you heavy into research. You know, you spent a lot of different time in research. It looked like you did some time in France and you did some time over in Spain. And then you finally jumped, I don't know if it's across the pond and then across the land, as they say, over to UC Davis before going to Berkeley. Lot of research, right? A lot of different research, a lot of different plant genetics. You come over to California, and I got to say, Angel, and I don't know where you were with cannabis prior to the jump, but you're not exactly breaking stereotype, Angel, that you rolled into California and immediately went, can I apply my brain to the cannabis industry, right? Was that something getting into it that you were familiar with before you came over, or did California just seep its indica into your veins?
That's exactly what happened. I came to California directly, as you said, to UC Davis to do my research in different crops. I was working with tomatoes, with lettuce, with forest species like sugar pine, redwoods, almonds, cherries. Always, obviously, at the scientific level and in academia. So I realized after 15 years of experience working poorly in research, I realized that the industry, that the real players, the farmers, the growers, the nurseries, they don't really get the science. In academia, we are very limited and very competitive to write papers, scientific articles. Most of them, 90%, they don't reach the audience of the consumers. So I say that is very sad that we are working in public universities and with the contribution, economically contribution of all the citizens, and they don't see the real application of the science that we do. So that's why I decided to start to fund MyFloraDNA. So the main goal is to bridge all the science and research that we do in academia to the industry and to the real players in the agriculture field that are the consumers, growers, and so on. So that's the main goal of MyFloraDNA, to bring this science.
Yeah, it makes me smile that it makes sense when you feel kind of like your hands are tied behind the back with all the actual work you're being done. You spend all this time with your research, you write the paper, you know, it's published, and yet three eyes end up seeing it. Whereas I take similarities with the personal finance industry. You have these economists who write these amazing papers, very technical, and explain and go in depth into what is happening and what they think. And then it's published with this 17-word title of like that no one's ever going to understand. And 4 people out of the country end up reading it. Whereas you have some schmuck like me on a podcast making financial advice, and yet I'm reaching tens and tens of thousands. You know, I find it neat that instead of going towards that research path, you're developing a company that brings a lot of those gains to the people, and being able to describe it to your customer is a pretty powerful thing and see immediate application to it.
That's correct.
It is one of those things, and Oscar Illan as well in there. So your job is working in the sales and marketing kind of part of this, Oscar, trying to communicate this out. You know, this is something, and we've had other both very testing heavy shows. Shout out to Dr. Kumar Bid. You know, we've had some people really centric on the growing aspect of it. And what we've seen is commonly call legacy people, people who are in this industry, people who are growing up in the mountains, people who are in the Emerald Triangle, people who are here and growing, and they don't need your fancy science to tell them how to grow some good weed. Those kind of people could learn so much from some basic information, let alone all of the information that you are able to provide at MyFloraDNA, but for just some basic, even if it's just cannabinoid or any sort of genetics based on there, not alone, not counting what diseases it is, Oscar, how have you found that issue difficult, that the embracement of these people who were here before testing became all the hot rage, right? Before testing and getting into the plant, before the young kind of scientific minded came in and said, hey, this is more than just something we grow. It's more than Mother Nature. It's more than the goddess ganja. Like there's this whole application of plant science to that. Have you found that message and getting that across and getting them to embrace the data and the results? Is that a difficult proposition for them, or what's it been like on your end, Oscar?
Well, for me, it was definitely a challenge, as Angel commented, to apply science to the cannabis industry, which was more on an infant stage about all these molecular genomics services that we can do. So it's a challenge because if I came from a different industry, I was always involved in business development, international business, import-export, and for me to understand how we do things at the lab level and the scientific level, and be able to translate that to the public, to the end consumer, I've been the buffer in the middle to be able to translate all that scientific, language to the end consumer, which is the breeder, the grower, the nurseries, and so it's worked pretty well. It's been a good thing right there, and I think that has allowed us to grow quite fast by translating the scientific language and culture to the more practical part of the business. Mm-hmm. Okay, especially to notice what is the pain of the market, what the market needs in the cannabis industry, and focus on that. Okay, and that's been very useful to put both worlds together and get the right services for our public. Okay.
Do you find that customers are coming to you because some regulation is forcing them to have to look into testing in general, or do you find that the customers that seek you out know specifically what you're doing and what you do very well, and that's why they're choosing you? I see regulation as almost being an open door for you. It's allowing the acquisition of the customer the very first time around to be a little bit easier, but then that's that point where you can differentiate yourself from all the other labs and facilities out there by applying these additional resources, by applying the science back end of this as well. But again, is it regulation that's bringing them to you, or is it your marketing?
Well, it's been regulation obviously help the industry, because as it gets regulated, the growers and the industry wants to offer a more professionalized service and added value to their product. So that definitely has helped us to keep the industry safer at the pathogen testing level. But from the grower's perspective of point of view, I mean, they want to have a clean and healthy crop. So, they're going to start doing it no matter if the regulation was already in store or not. So yeah, I mean, this is we cannot tell. We have clients. We're testing absolutely for everyone, not just for the regulated, non-regulated. So yeah, we're testing all across the states. In states where it's still not regulated, but they're working with CBD and other type of. So yeah, I wouldn't. It has definitely helped, but we would still be doing it if it wasn't regulated, I think.
And I think that's an interesting part about testing. And when you're creating a company, Angel, like MyFloraDNA, MyFloraDNA.com, and you're putting together what standards or what machines are you going to use, gas or high performance liquid chromatography. Their lack of standards in the cannabis industry, the lack of kind of federal regulations makes for some very subjective testing results based on the equipment that you use, based on the humans that are doing and using said equipment, based on the training that they have. How have you found that process diving into an industry where, as opposed to something like tomatoes or a more standard big ag vegetable or produce, there is a little bit more federal regulations? Our cannabis industry hasn't fully embraced that. And we have people who test at home and we have people who have these lab reports. In fact, I believe there's a lawsuit happening right now over the potency of THC. And as people get smarter and smarter at home and as technology gets cheaper and cheaper, there's going to be more home testing and they're going to have different results, but it's a different process. How has it been trying to determine, Angel, what equipment MyFloraDNA is going to use, or just trying to figure out where do you go when it comes to setting the standard, so to speak, and regulations and standard in the testing industry?
So I just wanted to make clear that we are not a chemical testing lab. Okay, we are not analyzing chemical or biochemical compounds. We are a molecular biology lab, a genomics lab, and we are always and exclusively working at the DNA level. This is our strength. Potentially, we could offer services at the chemical level, but again, there are many other labs in California and nationwide, so we don't want to compete with more labs. Our strength, our expertise is biotechnology, genomics, so we are focused on that. Concerning the standards, again, basically this is 15 years of experience working the same technologies in other crops. We are just extrapolating the same tools to the cannabis industries. Obviously, we are always adding new technologies, but molecular biology is not a tool that it was implemented the past 2-3 years. It has been around at least for the past 20-30 years. What happened? That only the large ag biotech companies such as Monsanto, Bayer, Syngenta, BASF, they had in-house actually the option to apply those technologies. And the small and medium companies, small, medium growers, they never had that opportunity. So that is our main role, to offer the same technology as the big players are using, offer that to the small and medium growers.
That's awesome.
So then if we take out, if we remove that chemical breakdown analysis from the company and we start thinking about the other product offerings that you are doing at a level, I'm wondering, like, are you developing a system that is helping to kind of give early warning signs that there may be issues or that there's issues with something going on with the genetics in the plant to begin with? But are you also speeding up that process for breeders? Are you able to break down and analyze something and specifically say that this specific trait applies to this in the plant, meaning this specific structure is going to bring out this effect in me, and so I can send you 10 plants to analyze and cut down my breeding time by a year? Are you able to get that in depth where you're specifically telling me, not only I saw a trait where you're saying this is the full genetic profile of this to kind of patent or trademark or show authority and authentication with, I'm actually saying on a deeper level, you're able to specifically pull out traits and say, this is going to have a high probability of inducing this?
Yes. So the answer is yes. So basically, we are working with what you are talking about. This process of this technology is called molecular breeding. Okay. What most of the breeders in cannabis and in other crops are doing is the traditional or the classical breeding. All right. So you have one plant, a male, female, you cross, and you get a new F1 population, and hybrids. And with the traditional and classical program, you need to plant all those baby plants, observe the phenotype, and by the end of the cycle, you will select which one is the winner. Okay, once you select the winner, you are
you're going back to start again.
Exactly. You will need to start again with the winner and start doing back crosses, and there are many strategies. But at the end, the process is going to take you 3-4 years. With molecular breeding, what we can tell the breeder at the first stage is which of those plants they are going to be the winners. So they don't need to wait 6-7 months. Basically, in 2 weeks, I can tell you, okay, so plant number 2, plant number 77, they have the genes that they are associated with the traits that you are looking for.
And my favorite part about this is that at every level of your customer, you're actually providing value. Meaning, if I'm a small breeder and I have just a couple patients as a caregiver to them, this does save me a year. And, you know, the time spent or labor cost may not be so much because I can still kind of manage with those patients in that regard. But when I think of a large MSO, to knock out 7 months and 100 employee salaries and all the benefits and the electricity, and it's like, heck yeah, for X amount per test, you're saving me a ton of time. That's a very powerful thing. Just even without tracking it, even without storing that data and trying to use that to make an argument in court or legality or authenticity, that just time-saving value is like amazing.
You can save time. You can save a space in the greenhouse, a space in the field. You can save labor. You can save inputs, electricity, water, and so on. So the benefits of molecular breeding versus classical breeding, it's incredible. So that's why only few companies they are applying these technologies. Imagine you are in Ohio, you want to come to California, you have 2 options. You can take a train, you can fly. So you're gonna reach the final destination, but it can take you either 1 month or 1 week, right? So it's the same. It's the same example.
It is. It's incredible. It's just incredible to me sometimes what, you know, we continue to find advancement. And I'm assuming you may have written this, Oscar. Like, I came across Angel and learned about him, I think from a posting that was talking about, and I'm gonna go on this. I wanted to ask you how to say it, but I would like to be brave here. I'm gonna go with hop latent viroid. Is that correct?
That's correct.
Is it viroid or viroid? I was really trying to struggle what that last word was in that.
I think it's viroid, but
viroid's a virus that makes hop latent viroid. So I came across an article, or maybe it was on LinkedIn, with Angel Fernandez from MyFloraDNA.com, talking about hop latent viroid and kind of what it is. And I think this is one of these things we're talking about. Caught my attention as both a IPA consumer. I like hoppy beverages as well as cannabis, which I've learned I just learned this recently, how close the hop is to the cannabis in the structure of it or something. I'm probably, once again, not as smart as all the people on screen with me right now. So let's start there, Oscar. The hop latent viroid kind of, you know, press release and information came out a little bit about that, either from both of you, like what that is one of the few, or one of the many things you guys are testing for there. But I was shocked to learn you got some amazing stats on there about how many people are
scary
plants projected with this, which are basically starting with stunted growth, doomed to be stunted grown in many other kind of deficiency plants, in something, as we just learned, could be prevented if you realized, and you could be treating for HLV, right?
Right. Well, I mean, yes, hop latent viroid has been in the industry for a long time, okay, but just in the last few years there's been a lot of available testing to do it. I think we have developed probably one of the most accurate testing in the industry at probably the most competitive price, and that has allowed and also as well the process, the collection protocol we have narrowed down to a very simple process, so the grower or the nursery can send us like many thousands of samples if they want to within a short period of collection time. Okay, and that has helped us a lot to help the industry quite a bit. Okay, Yeah, this is the reason why hopulating viroid is the most devastating pathogen right now, because it's a viroid that you have to do constant screening. I mean, some viroid means that some parts of the plant might be infected and other plants might be okay. So that forces you to do like constant screening from time to time to make sure that plant is not infected. Our protocol is quite simple. We normally take like 3 or 4 parts of the plant to test. And that way we make sure that if the plant is infected, we're going to detect it hopefully the first time, right? In regards to go ahead if you want to ask anything.
No, so I'm still trying to understand it because I'm an idiot. It like the difference between a virus and a viroid. You know, is it that the virus is in the genetics itself? It's already in, and the viroid is something that is kind of happening after the fact on like a cellular level in a different way.
A virus will be all over the plant. A virus, but a viroid will be only in certain parts of the plant, kind of like a herpes on whatever. So
yeah, so
it's the same way. Yeah.
So then, as it applies to like testing and stuff like that, or say I'm setting up clones or I'm doing tissue cultures, is there a way that I can, at the initial stage, prevent this from the beginning? Like we test with you, we know that's all right. Then my tissue cultures are going to be fine,
Ideally, we would obviously we would recommend to test any mother plant you're going to be cloning from. That would be ideal. But then again, in some cases that might be like a pretty big budget. So basically, what we try to do by adjusting our prices as much as possible is to help the industry to do as much screening as possible. Okay, so I mean, if you ask me how much screening should I do, I tell you all the mother plants. I mean, everything you're going to be cloning from, I will recommend you to test before you start like cloning the plant. Okay, but then I understand the needs of each client, and we adapt the protocol.
And how does that work with like protocols are great, but still transferring of tools? It's like it's every bit of their process. That's what's going to maintain that quality. And to me, it also, I mean, let's just cut to the chase here. It seems like a pretty awesome business model to be in if there's that much worry to this happening, and it's that easy to transfer this between things, and there is this huge swath of the country that is having this problem and don't even know it, which then just seems like it's going to make things even easier to kind of work through. At what point do you see yourself in the industry stopping a lot of this? Like you guys are like the only ones I saw that are doing a lot of this. So like, and that way you're trailbreakers or trailblazers. I mean, like, how do you feel being the only one screaming about it?
Dustin, I will tell you that I would prefer doing DNA analysis for breeders that they want to discover new genes and they want to breed new strains rather than doing the pathogen testing. If we are doing pathogen testing, it's part of our business. It's just because it's a real need, and our main goal is always help the industry, right? But as a scientist, I would prefer spend more time and resources doing research, discovering new genes, and helping to create new strains, more sustainable, better quality, and blah 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 blah.
It's I was gonna say it's still just pretty incredible to me sometimes, kind of the lack of, I guess, adaptation in our industry. So do you think where you're building towards is the growers getting better at making super plants? I know that sounds really silly, but I feel like the future of this, it starts with preventing badness in the beginning, right? We've seen the sci-fi movie many times, and now we turn our forgotten, and now we're just basically making super plants. Essentially, is this what this leads to? Things like CRISPR and such, where we're just making on a molecular level like a magnificent growing plant that can survive in really rough regions and temperatures and still be able to grow in cold weather? I mean, is that kind of the future application of what this research is leading to?
that is going to be the future in plant breeding in agriculture, and we will see that in 5, 10, 20 years or even earlier. But super plants, I mean, you as a consumer, you are trying to get always the best fruit or the best plant that you can. You as a breeder, you are trying to use less inputs and spend less time as before. And if you are a farmer, you are trying to also to reduce the application of pesticides, fertilizers, so on, right? So you put all those things together. Right now, the best technology is CRISPR and molecular biology, molecular breeding and CRISPR. And coming back to the same example as before, if you want to go from Ohio to California, you can take a small plane or you can take a super fast plane that instead of 5 hours, you can do that in 30 minutes. So it's the same thing. The main difference of CRISPR or gene editing tools versus GMOs is that with GMOs, for example, if you were trying to create a tomato resistant to a specific disease or pathogen, you could take a gene from lettuce or from an eggplant and transfer that gene to a tomato plant. All right. So you are working with 2 different plant species. With CRISPR or gene editing, you are working at the same plant level. So you are just knocking out or knocking in genes from the same plant. So it's like you just turn off the light in one bedroom and or turn on or turn off, but you are always working with genes that they are present in cannabis. So you are not transferring genetic material from other species. So that's why this technology is becoming very, 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 very popular, and it's going to be obviously not only in agriculture, but in animals and in human health. Right now,
besides comfort, I think, is there one reason why anyone would be better? One seems like people have an inclination to be more accepting of CRISPR. When you're saying it's all coming from the same genetic base, you know, they're like, oh, that feels safe to me. And when you say I'm sticking an eggplant into my cannabis plant, they're like, oh no, not this crazy mutant plant. You know, is there truly any better way between them, or is it just another way of applying a different genetic? You can just do a little bit more with a different system because you can pull traits off in a different way.
It's marketing, Dustin. It's marketing. Scientists in general, they don't know how to market. They don't know how to reach the public audience. I can tell you that 90%, I would say 95, but let's be a little bit conservative. 90% of all the geneticists and scientists worldwide, they are pro with GMOs. But 10, 15 years ago, they didn't know how to, or 20 years ago, they didn't know how to reach the audience. And so with this, and obviously it didn't work, right? So with this technology, now there is a consensus at the scientific level. There are many important universities led by UC Berkeley, MIT, and many more, trying to avoid the same mistake and improve the communications between academia and the general public. And the most important is that with this technology, CRISPR, we can cure and save many lives. So agriculture obviously is a very important field, but if you do a little bit of research about the application of CRISPR in humans and in medicine, it's incredible. It can cure so many diseases that's why there is a lot of interest, investors, many departments in universities working with that. So But again, it's gonna be. I think the battle is gonna be if scientists in general they are gonna be capable or able to translate and communicate properly the applications of this technology.
I have nothing. I am super happy to have learned what GMO versus gene editing meant. Like I never knew that difference. So I appreciate you just explaining that to me. Speaking of which, Oscar, let's explain to them. I know we want to talk a little bit what MyFloraDNA does and maybe look at a certificate that we receive. So let's talk the process, because I'm looking on the website, MyFloraDNA.com, and it says how you're based in California and you can go to California and you arrange pickups and such if you're in that state. But it talks about mailing in samples and such. And I was curious, just is that actual THC that people can mail in? How does that work?
Well, basically, most of our clients are based on the California area, which mailing we don't mail plants. We just mail like a bunch of tissue in, like, let me show you real quick.
I get you. All right. So is that
there's half an inch of tissue in these tubes. Okay, we're not mailing the plant. We're not mailing like leaf tissue like in the in the box. Okay, so we haven't had any problems so far with mailing like tissue.
The cotyledon. Normally, our clients they mail the cotyledon, which is the first part of the plant that doesn't contain THC.
I was going to say, I think it's like similar to the recent ruling that came out where the seed itself, because it contains less than 0.3% delta 9 THC, that it technically doesn't count, to that legal loophole where all these other cannabinoids. So because they're sending genetic material, essentially, they're always going to be less than that 0.3% D9 THC threshold. So similar to mailing seeds, which we've already talked about on past shows and such, like that is where the legality. So they can mail you the genetic tissue. They go on the site, they order the test, they mail you the tissue. How does that process work then, as far as what MyFloraDNA.com does?
Yes, as soon as we receive the samples from our clients, we previously send them a collection box, and we create a dashboard for them. We call it MyFlora Cloud Dashboard, where they can track and follow up all the results we do with them, especially. 90% of our clients are recurrent, so they see that dashboard very useful. They can do statistics, find the invoices, and an educational area where we upload a lot of ebooks and posts that we do about the pathogens and any other molecular discoveries that we do. Okay. After that, after we receive the samples, we're pretty fast with the turnaround time. We're doing about for about 1,000 samples, we're able to do within 24, 48 hours for the results. So that's quite convenient, because the earlier you know about if a plant is infected or not, the better. That way you can just go ahead and pull the infected plants away. So the whole process is fairly fast. Within a week, you're going to be able to know if from the moment we send you the box, the moment we receive the box, it's going to be about a week. You're going to know the result of what we do in the lab, right? So it's quite easy, convenient, and fast. Okay.
I think that's what. Let's take a look at. I think you had some sample results here. Let's see if we can pull that up. Let's see. Let's add you on screen here to that. This is talking about the molecular ID. Do you have the sample certificate of analysis? All right, essentially there.
Of an analysis.
Yes. Or we can talk about this. I apologize.
Which has a different type of analysis, but still, the dashboard may work similar and just have a different lab result. Correct.
Correct. The company, our company does on one part we do like pathogen testing, like HLV, botrytis, fusarium, and all the other pathogens in the industry. But we also recently developed a fingerprinting technology, which is a patent pending way to show, it's a visual way to show the social security number of your plant, okay, which consists in almost 3,000 SNPs of your plant. So what you're seeing right here will be like your plant genetics, okay. So every one of these spots right here is like a SNP of your plant, okay. So this is very good to do for the validation purposes of the plant. So with this type of a certificate, you're able to contrast your plant with any other plants in the industry. And if you want to give that extra trust to your clients, you're able to give this certificate to them. All right. So yeah, the way we do it is like, once we do the molecular ID certificate of your plant, which is going to be unique for your plant, we're going to be able to contrast any other plants or any other tissue with the plant you already tested with us. Okay, that way you give that extra trust to your client that what you're selling is actually what you're saying you're selling, and the other way around, the consumers are able to double check that what they're getting is actually what they're buying for. Okay.
So this technology has many applications. So for example, the certificate,
I can name 7, yeah, at least 7.
At least 7, and because we stop mentioning them. But for example, if you are a breeder and you develop a new unique strain and you want to have the certificate and to protect your strain, we highly recommend to get this passport number, social security number, or molecular ID based on molecular markers. When Oscar was mentioning SNPs, SNP means a molecular marker. So we are analyzing, let's say, many genes or molecular markers that are in your body, and they are unique to you. So that means that when we put that in a paper, those molecular markers are going to be obviously different to the molecular markers that we are analyzing from BC. So when we compare BC molecular markers versus Dustin molecular markers,
yes,
obviously we're going to be able to say, oh, those two plants, they are different.
Okay. But let me ask you a karaoke gene. That's what we learned. I got the musical pipes, fellas. That's what we missed. That's where we're going. Let's bring it back around. Sorry, Dustin.
In my head, I'm thinking about this in 3 ways. How does this provide me security against larger corporations like Monsanto or something, who are doing utility patents on the genetics, and I, as a small breeder, don't really have any way to really, this is, in what way can I take this and apply it to someone else? Unless they are scanned into your system, you don't necessarily know their genetic marker. There's a lot of claims that can be made outside of just you. If a claim is made, then what? I got to get a sample, send it to you, and you'll tell me, compare it against the grid. The secondary effect of that is, like putting that to usage. If 2 separate companies are doing the same thing and we both apply our trademarks, both do our testing and have this sitting on 2 separate ledgers, what is to say the difference, and what really doesn't change the genetics as it applies to soil and other things that happen is there any, can there be a variance after the fact from one breeder to the other who used the same tissue culture?
So, I mean, that is a question for a patent attorney. I guess that the winner would be the first person or company who applies for the patent. What is true is that before or even currently, when you want to register a new strain or cultivar, no matter which crop, to the USPTO, you just had to provide morphological data or phenotypical data, blooming season, THC content, or kind of agronomical information. Right now, they are accepting, and by 2025 or 2026, it's going to be mandatory that every time that you want to register a new plant, it needs to include a molecular profile. All right, so that is the only way to be 100% sure that this is very unique.
Where does that start? Who actually is saying that the guy who is testing cheese UK, has actually had it to begin with? Who actually designates base plant number 1 into the system to prove the genetics after the fact?
I mean, again, I'm not a patent attorney, but in case that there is a conflict, they will ask for proof. So probably you will have to go to your lab or to your greenhouse book and saw that you have been doing this cross since 2017. I don't know again, but I guess that they are
interesting for sure.
But also the good thing right now we have is the blockchain. So, for example,
I love it.
And that is going to be super helpful, because if you are a breeder today, you are developing this strain, you generate this molecular ID, and once you get it, you can upload it into blockchains that no one ever can change it. That is going to be a very strong proof to demonstrate in front of the judge that you are the first creator, let's say.
Yes, and depending on how you're building your system out, I mean, getting that data off the chain to be put to use in another system is not that difficult. You're fully capable with the technology as it stands today. So as we think about you integrating in with other molecular profiles from other systems that can at least match quality or match certain attributes in the same way, I think it's going to prove to be immensely efficient cost-wise for a breeder to use a system like the blockchain in order to manage this. You know, now I don't need an extra staff and paying this person for a whole year's salary to get these things on the chain, to get the tracking started, to see it along the line. As I'm working the plant, I could be updating the ledger or updating the genetics as we're doing stuff, you know, new things with them. It's pretty powerful. I find that you're even thinking in that forward level. You're the first company out of 40 that we've had on the show that is blockchain forward in their thinking. And to me, this is 12 years. This is like 2007. I've been dreaming and feeding stuff like this. So it's like, it matters.
As I said at the beginning, Dustin, our main goal is to help small and medium breeders and companies. And with this technology, is super helpful to protect breeders. Every time that you have a new strain, for a few dollars, you can get your certificate
with a cryptographic timestamp as well. You know, it's
basically we created a tool for many platforms already in the industry that are working on protecting the IP of the different cultivars. We're just offering a tool, a genetic tool, to give extra support, extra added value to what you have. You know, having a code, a metric code, whatever code, is not real proof and evidence that that's your plant. The only real proof and evidence that's your plant is the DNA extraction of the plant and the genetic profile of your plant. That's what we're doing. So we're only offering the tool. I mean, we're not the judge of who's doing what. I mean, we're just telling you, hey, this is what you have.
And that's the best part about what you do, though.
Yeah.
I just think it's going to be some of the just what's going to come out of this, right? Both whether it is, you know, just getting into the gene editing and creating these kind of terrible term, but super plants, like these genetically resistant plants, which we're already working on, but just what some of the further advancements can go. Obviously, you know, the idea of creating and protecting strains on a molecular level and cultivars and processes is fascinating. I just think it leads to, obviously, just in this short discussion, we can see some of the potential, both excitement and some of the negative aspects of it. It's going to be really interesting as that goes forward. I saw something else on MyFloraDNA.com for both of you. You also have a cannabinoid dominant testing service on there as well that's looking to see where plants are. You know, we just talked about being able to mail genetic material, you know, that 0.3%, which may go up to 1%, 2023 Farm Bill, shout out if it passes. So they're looking to raise that to 1%, but right now it's below that 0.3% for industrial hemp, CBD, and all these other things. So you're able to, on a genetic level, pretty much, I guess, explain to me then how that is on a genetic level, determine what its actual potency is going to end up being, because I feel like there's so many other outside factors and growing environment and such, in either cross-population or cross-pollination, I should say. How do we, on a molecular level, determine kind of what that actual THC percentage is, or marijuana versus hemp?
It's just based on presence or absence of genes. So if the gene is present, that means that it's going to be high THC. We cannot tell you if it's going to be 5% or 70%, but we're going to tell you that it's above the threshold of 0.3. If the gene is absent, it's going to be below 0.3. So that information is extremely very helpful to those breeders, that, in particular hemp breeders, that they are planting thousands of plants, and they don't want to wait 16 weeks. So basically, in 1 week, we can tell them if they are below or above 0.3%. We have heard many cases that after growers that they have been growing plants, then they realized that they were above 0.3 and they were confiscated by the...
Yeah. Exactly. Or the whole field has been ripped down. You know, you have 10 acres or 100 acres that's gone. And I wonder, you know, I kind of find if this is just another tool in my toolbox to use as we're continuing on. You know, I'm thinking, BC, we had a good conversation with Greg Selby from Science LED about the light being able to pull off specific traits we're looking for. When I think about using this tool that tells me this plant's disposition, this genetic disposition to be this way, and then I couple that with another tool like a special LED system that caters out specifically those things, we're really looking towards a bright future, a more insured future as an operator, as a company who has to do this for money, who has to make a living and pay for my kids' dance lessons and stuff like that. I would like some more assurances as we go along with these processes. And that's kind of what your tools are giving us. It's an extra insurance plan for very little amounts of money when you consider that. And I find it powerful as a value all the way around.
It's exactly that. It's an extra insurance. It's a prediction of how your plant will look like. So you put those things together, you can save time, money, many. Has many advantages.
I mean, you guys are...
I was going to ask real quick, Dustin, before we go, because I think it's pretty exciting. I was reading where you guys are located at in AgStart, which is kind of this agricultural startup with all these other different kind of labs and such kind of together. And they got a cool little video on, it's both on MyFloraDNAs.com as well as AgStart's website. So what was that like? What's that like being around not just yourself in your own laboratory, but you have this, I guess vibe is the best way I can put it, where every floor in your building is someone that's working on cutting edge technology, wet labs, dry labs, all these different things. I would think that's pretty exciting to be not just what you guys are doing, but you have all these other, and I think several of them are breeder and cannabis specific things working on you are like super cutting edge cannabis office headquarters there. What is that like?
I was going to ask them the question of like, what are they looking towards with excitement towards the future? And it's like, they're on the bleeding edge of everything. Like
everybody is the future.
Exactly.
Yeah, it's all actually it's all about innovation. Everyone that working in this lab, you know, it's all like brilliant scientists, like working on very innovative stuff. And the good thing about it is like we all share the knowledge, and we all share, besides the knowledge, we share a lot of equipment to make the lab as complete and brilliant as possible. And you know, we help each other a lot because the community here, I mean, as you said, we are surrounded. Our whole idea is to grow and be innovative. So we're always helping each other, and we're in a great place to actually, you know, build up new services and new innovations. So we're very happy to be here. And this has actually allowed us to grow a lot faster than it would have been otherwise. Okay.
It's always crazy, some of the cutting edge things that are happening. You guys are on the cutting edge. There was actually just a recent report. You may not have even seen this. It just came across. Ethan Russo, MD, along with a few other doctors out of Portland, Oregon, have decided or published this paper just right before we went live, though this will be a couple weeks from there. Aroma of the cannabis plant plays more into the user's effect of it than the actual maybe THC. I know right now where we deal with in our growers, everything's terpenes. Terpenes is the magic word. I must hear the word terpenes about 95 times a day. Like we're getting away from sativa and indica, bro. We're talking about terps right now and all these things. So we get into that. And now, like just recently, we're learning maybe it's just the smell, partly caused by the terpenes that is coming into that. What do you think, just in general, what do you guys think about that? We just learned about that right before we hit record on the show. I just introduced it to Dustin, and Dustin said, well, foods kind of learn that same thing. Like a delicious cherry pie is just as much about the smell. Is that, how do you guys feel about that study that came out?
I haven't read that paper. I will do it hopefully tonight. But it's true that terpenes, aromas, they are all also controlled genetically. I have been working in, for example, in peach at the aroma level and determining nutritional traits, vitamin C, phenolic compounds, and things like that, and we have always found connection or genetic variability and genetic association with those traits. So yeah, I can't wait to read the paper and see what it talks about.
It always excites me, Dustin. I always love having guests on, Angel Fernandez and Oscar Illan from MyFloraDNA.com, MyFloraDNA.com, link to it in the description below. I just love talking about the future. I both fear the future, right? As I get older, we just learned before we went, Oscar and I, same age, both of us still doing pretty well. I'd say, Oscar, if I'm being so humble myself. I just love where the future is headed. I love where the future is going. Do you have anything, Oscar or Angel, about the future maybe in the cannabis industry or testing or something? I know we should give a quick shout out. We can't let Angel go without. Everyone should check out the almond tree genome, right? Part of the compendium of plant genomes. I also learned what sea buckthorn was today, by the way, just for you, Angel. I Googled. I have no idea what sea buckthorn was. What about the future excites you? Just in testing and CRISPR? I mean, I feel like, as Dustin said, that's coming out now, but it's already behind the scenes in Sweden. It's so 2017. What is the future? What excites you guys?
So I think that if society in general let the scientists to work, the future, at least in biotechnology, is going to be CRISPR. They are going to be able to save many lives, to make new medication more efficient and more productive. And in terms of agriculture, the same thing. We are all scientists and companies trying to improve plants more sustainable, with more yield, resistant to drought. So I think that we all have the same target goals, and we will see in 5-10 years where we are at. Reduce climate change. Yes. Yeah.
No, and as you said about the terpenes and the display, this is great, because as cannabis gets more legalized, people are getting more educated. Okay, it's not all about THC. Okay, it's about terpenes, about the flavors, about many other things, kind of like the wine tasting. Now cannabis is not about, oh man, this is such a high THC level. This is great. No, it's not about the THC level. It's about many other things, many other factors that influence the quality of the cannabis. So I think it's a very positive thing. The more education, the more educated we are, the better. And in the future, it's a great future. As it gets more legalized, we're gonna get a bit more educated, and hopefully, we'll be able to grow and offer our services all over the world. That will be our idea.
That is what we are looking forward to. Thank you, Angel and Oscar from MyFloraDNA, MyFloraDNA.com. Thank you for joining us on today's show. Thank you for really helping us learn a lot about what molecular testing is, the difference between GNO and gene editing, and more. I thought it was really exciting. Thank you both for being on. I appreciate you, explain it to me like I five. That's how the way I'm going to break the fourth wall. They said, what do you want to talk about? I said, just don't make it too smart, because sometimes our guests get too smart for me. I am not at that level, though. So thank you for joining us and really helping educate both Dustin and I and our audience. I really appreciate your time today.
Thank you.
60 minutes spending with us.
Thank you very much for having us. Thank you.
All right. Thank you. All right, everyone. Be sure to join us every single Friday, 4:20 p.m. Wayward.media. Wayward.media. Check us out at Meet WM. That is the place. Also, audio. Spotify, Apple, iTunes, Amazon Music, Google Play, Stitcher, pick it, name it. That's where you're going to join us. Follow, subscribe, click that button. Tell them, Dustin, click the button, subscribe. We'd appreciate it. Check out MyFloraDNA for your testing needs. Have a great week, everyone. We'll see you next Friday.
Please click the button.